Hi, everyone. Thank you so much for joining us today. We will be starting the webinar in just a couple of minutes. But first, we'd like to present to you just some guidelines and the team. So my name is Alina Grenier Arellano, and I work with Jay Kumar and OSPF, and we're so glad to be bringing you Christopher Austin, who will be talking about drug repurposing and its impacts for our research agenda, especially in these unprecedented times for present and future pandemics. So just a little bit of information for you to make this as easy as possible. Right now it's being recorded, but that will be recorded is essentially Christopher Austin's speech. You are not on video and you have all been muted. If you have any questions for Dr. Austin, please put them in the Q&A, which is at the bottom of the Zoom screen under Q&A. And if you have any questions or want to ask me about technical issues, please do it through the chat box, which is at the bottom left. We are welcoming more than 100 to 150 people from all around the globe. So please share with us your just some answers that we've put in the polls that you must have seen when coming online. And they're just a little bit of insight we want to know about you so that we can know better how to present these sessions going forward. Without further ado, I am going to I'm going to hand it over to Jay to present Christopher Austin. Thank you.
Thank you so much, Alina. If you could open up my video, that would be great. We are very pleased to be here today. I'd like to welcome you to the latest in a series of events and dialogues hosted by the Open Source Pharma Foundation, which is dedicated to using open source principles to create a world of medicine for all. In this epoch of the COVID-19 pandemic, much of humanity is thinking about the same few questions. How can we create medicines and vaccines? How can we create them more rapidly? And will they be accessible to all? As background, in the standard drug discovery approach per Tufts University, we need 12 to 17 years to develop a single new drug. In this time of COVID, we don't have that long to wait. Today's topic of repurposing existing drugs thus becomes particularly timely, not just because of speed, but also in terms of access. It's our pleasure, therefore, to introduce you today to Dr. Christopher P. Austin, a true pioneer in improving the system of translational science and drug discovery. Chris heads NCATS, the National Center of Advancing Translational Sciences at the National Institutes of Health in the US government, which is, at least to my mind, the lead body for drug discovery for the US government. Chris's background is impressive and stellar, and it's a great privilege for us to have him here today. Chris leads NCATS, NIH NCATS' center's work to improve the translations of observations in the laboratory, clinic, and community into interventions that reach and benefit patients, from diagnostics and therapeutics to medical procedures and behavioral changes. Under his direction, NCATS researchers and collaborators are developing new technologies, resources,
I think Jay Kumar's speech was cut short, but essentially we're very proud to be bringing Dr. Austin, and his career has spanned the spectrum of translational research in the public and private sectors, and he joined NIH in 2002 as the senior advisor to the director of translational research at the National Human Genome Research Institute, the NHGRI. He was appointed to the NCATS director in 2012 before joining the NIH. Austin also worked at the pharmaceutical company Merck, where he directed programs on genome-based discovery on novel targets and drugs, with a particular focus on treatments for schizophrenia and Alzheimer's disease. Austin is trained as a clinician and a geneticist, and he is a member of the National Academy of Medicine, formerly the Institute of Medicine. He earned an MD from Harvard Medical School and an AB summa cum laude in biology from Princeton University. He completed a research fellowship in developmental neurogenetics at Harvard, studying genetic and environmental influences on stem cell fate determination. Austin also trained in internal medicine and neurology at the Massachusetts General Hospital in Boston, after which he practiced medicine in academic and community hospitals, providing primary care in urban settings and in rural Alaska and Africa. So without further ado, thank you so much, Dr. Austin, for being with us. We're very lucky to get your insights, and I will hand it over to you. Thank you.
Thanks. Well, thank you, Lena. Thank you to Jay Kumar, wherever you went off into the ether. I'm actually talking to you from the office today. I'm not usually in the office. I'm staying home, working from home, probably as most of you are, but I don't trust internet connections except from the office. So that's why I'm here. So it's a real pleasure to be here. I've gotten to know the Open Source Pharma Foundation and Jay Kumar quite well over the last several years, and find that they are really fellow travelers. You are really fellow travelers with us here at NCATS in trying to remake how the therapeutic development and deployment process works, or in many cases doesn't work for the people who need it. And you know, Jay mentioned that in the era of COVID, people are aware that we don't have time to wait for the 15 years that it normally takes to make a drug. But all of us on this call, I think, know that that is not unique to COVID. It's simply that our populations, or the publics, I think for the first time, for most of them, realize what it's like to have a life-threatening illness and not be able to wait for the current creaky, expensive, failure-prone process to reach them. And so it's and Jay and I and the other open source pharma folks have been talking about, among other things, the potential for a global open source repurposing hub. It's just and Jay and I may want to talk about that in the Q&A. This is something that we've been talking about. NCATS has been talking about with the ATRIS, who are our European counterparts, as well as the open source folks. So, so what I'm going to do today is go through some information that I hope will be useful, and I hope also thought-provoking. And you know, once Jay and I get talking, we have a tendency to go on for hours. So Alina, you will have to stop us in an hour. But I just want to reiterate what a pleasure it is for me to be here. You know, I guess the one other thing I would say to start is that I personally, and NCATS in general, feel very strongly that the translational problem is a worldwide problem. It is not a US problem, it is not a European problem, or an Indian problem, or a Japanese problem, or a Chinese problem, or what it is a problem which spans the globe. I have spent a considerable part of my directorship of NCATS working on international collaboratives, and continue to, because I find such value in them. I also should mention, and I think a lot of you will appreciate this, that this can be a lonely road, what we are all engaged in. And so I don't think we should shy away from the fact that having fellow travelers to boost our confidence and have at the least folks to gripe with about the problems, and at most folks to share insights with, is really important. I mean, I hope this movement will become more widespread, but for now, I think we have to stick together, those of us who are interested in this throughout the world. So it's great to be here. Okay, so what I'm going to do, I'm going to try to share my screen now. host as disabled participant screen sharing. So Alina has disabled me, but I have a feeling she will enable me here in a moment.
There you go. I think it should work now.
Got it. Okay. Where am I? Of course, it would help if I opened my, sorry about this. Here we go. Close this, open this. You'd think I'd be more, okay, here we go. You can see that, right? Yeah.
Thank you.
Okay. All right. So I've titled this Drug Repurposing in the Open Source Model. And I am going to go back and forth. You know, drug repurposing is certainly not the only aspect of the open source model. I don't need to tell you that, but it does have some interesting lessons and applicability to this space. So first of all, I want you to know how I conceptualize this problem that we are all engaged in, that I view this time in scientific and medical in the scientific and medical world as really a Dickensian moment. It really is the best of times in some ways on a scientific level, and it's a quite painful time, frankly, on a medical level because of the discordance in progress that we've seen between science and medicine during my professional lifetime in the last 30 years. If you think about the advances in fundamental science that have happened in the last 30, 40 years, they really are epical. You know, the list of things from PCR to CRISPR to MRI to you know, you name it, the Genome Project, what graduate students can do in the lab now in a day is what we used to do in the lab in 4 or 5 years, and that really is miraculous. What you're seeing there is a Genome Project, of course, and those are stem cells with differentiating into neurons. And that's all wonderful. But on the right side, if you ask, well, how is health doing? How you think about this personally, the last time you were at a doctor, you went to your primary care physician. I hope you've been going to your primary care physician. The last time you were there, how different was the experience than what it was 10, 20, 30 years ago? For most of us, the experience is not very different. In my own field of neurology, very few of the diseases that I used to see were treatable back when I trained. And the same is true now. Nothing has changed for most of those patients, even though the science has advanced in unprecedented ways. So it's an interesting question for us. Why has this happened? You know, it's a truism, an article of faith in the scientific and medical community that that medical advances will happen on the back of fundamental insights. But if you have fundamental insights that have increased by 4 orders of magnitude, and medical ones that have maybe improved in some ways by 1 order of magnitude, that discrepancy, that gap, has only gotten wider in the last 30 or 40 years, and we see this in the poor transition of basic or clinical observations into interventions that tangibly improve human health and get to the communities that need them. Those interventions are even when they are successful, because the development is so failure prone and inefficient, they tend to be very, very costly, because if you have to do something 1,000 times in order to have one success, then you got to multiply the unit cost by 1,000. And that's one of the reasons, not the only reason, but it's one of the reasons that these have become, interventions have become so expensive and out of reach for much of the world's population. But I don't need to tell you, this group, that it's very clear that even when we have interventions that work, everything from drugs to behavioral interventions, we are very poor, and by we, I mean the community at large, very poor at getting people to do them, or getting the drugs to the people who need them, demonstrably useful interventions. So the pain that I feel in this space as a physician it tempers my excitement as a scientist. That is, that there is enormous opportunity here to deliver on the promise of science to patients, but I am painfully aware that we are not doing a very good job at that by almost any measure. So at NCATS, our approach to this is head-on. Our organization was started a little less than 10 years ago by the NIH because of this realization, and the realization that we need to bring science to this process and to rethink on an experimental level every scientific and operational question that underlies this therapeutic development and deployment process. And what we really are focused on is developing new ways of doing things, innovative methods, technologies, paradigms. The open source paradigm is exactly the sort of thing that we like to work on. I mean, if you think about the cause that the translational failure when a fundamental discovery doesn't make it to people who need them, roughly half the time, that's because of a scientific limitation. Biology, chemistry, physics just doesn't work. But about half the time, it's not hard science. It's what we might call social science or soft science. It's access, it's ethics, it's IP, it's credit, it's collaborative structures, it's sharing, is these things that are human behaviors. And I go back and forth trying to figure out whether I think it's harder to figure out how Mother Nature works. That's the biology, chemistry, physics part, or it's harder to make human behavior change. I'll let you make that decision on your own level, but I think it's a close competition, and perhaps the latter even harder than the former. But that's what we're about. And I hope you know what we mean by translation. We've been using it in context, so I hope you know what this all means. There's definitions on our website if you want to see them, but you may not be familiar with the way we think about translational science. Our hypothesis here about how we're going to change the rules of the game here with regard to this process is by understanding the underlying science, and by that I mean biology, chemistry, physics, as well as the operational side, which underlies this process. If you think about this process, and you all work in it to some degree or another, I'm sure all of you, it's a remarkably empirical process, trial and error process. We really don't understand the fundamental principles of most of what we do, and therefore it's a trial and error, mainly error process. And so our goal is to turn this translational process from just that, a phenomenological trial and error process, to a predictive science. Sometimes I like to say it, I feel like, as I've spent my whole career in this field, and I feel sometimes like it's more like astrology than engineering. And so that's the transition we want to make. And when I think about my own field of genetics, that field has undergone that transition since the 1950s. It was a really empirical field. You think about Mendel and his peas years ago, and now it's completely predictive because we understand, not completely, but largely predictive because we understand what the fundamental rules are. That's what we're trying to do here. Okay, so now let's talk about repurposing here. So if you think about a therapeutic development, there's essentially two approaches. And I made this slide back in 2008, but it's still true now. You've all seen this chevron diagram, I'm sure, going from a target to a screen to a hit to a lead op, to preclinical development, clinical trials. We do all of this stuff at NCATS, and a lot of you probably do too. I put FDA approved here, but it could be, it could be the EMA or the Japanese Pharmaceutical Development Agency, or doesn't matter, and whatever the national regulatory agency is. And the conventional route, at least that I learned when I was in pharma, is because we don't understand what the rules here, you just got to screen a ton of compounds. You just got to take a whole bunch of compounds, 500,000, a million, 2 million, whatever it is. You screen them across, an assay that's a model for a disease. And it takes you, as Jay said, somewhere between 10 and 15 years to go from all of those possibilities to a drug. And it fails about 99% of the time and costs around $2 to $10 billion, depending on how you do the math from the beginning to the end. And so we and others a few years ago had a very simplistic thought that, well, gosh, what if you took every drug ever approved for human use we start out with major markets, so FDA, EMA, Japan, Canada, and you put them in a collection and you screen them. First of all, how many are there? There's only about 3,300 of these things, and we say by drugs, I mean small molecule drugs. I'm not talking about proteins or biologics for technical reasons here, but about 3,300 drugs. And the idea was, if you could put these in a screen, that is a testing system that was relevant to a disease, you could hypothetically skip over that lead out, preclinical development, all of that stuff, and go right into clinical trials in a year or two. So it's very very tantalizing. However, the fact that I was using that slide in 2008 and I am still using that slide now should suggest to you that it's not so simple. So this whole process does seem remarkably simple, but it hasn't actually borne out yet. And so the question that NCATS started to ask, has been asking for a number of years now, is why? And that's something that we want to talk about today. So, but I want to give you a sort of conceptual framework by which to think about this broadly before we narrow down. So there are about 7,000 diseases that affect the human family. About 95% of those diseases, so roughly 6,500 diseases, have no regulatory approved treatment at all. So most of the diseases that I treated when I was a practicing neurologist would fit into this category, but you probably all have your favorite disease. So there's two approaches to this. One is to find a drug that's currently approved and repurpose it, that is, find a new use, or to develop a new medical entity, an NME. That's a new drug of some sort. And I did a calculation a few years ago about how long it would take at the current rate of progress to develop a new medical entity, a new chemical entity, a new medical entity for every one of those untreatable drugs. And the number is quite shocking. At the current rate of development, it will be over 2,000 years before every human disease is treatable, has a regulatory approved treatment. So I would propose to you that that is not an acceptable answer. That we've got to find a fundamentally different way to solve this problem. So one of the approaches is to say, well, gosh, could we do an experiment and ask, well, of those there's 3,300 drugs that are approved already, and there's 6,000 diseases. So it's an interesting question to ask, what percentage of currently untreatable diseases could be ameliorated, maybe not cured, but treated to some degree by a drug in the current pharmacopoeia. That means the collection of approved drugs, and that's something that we're interested in asking at NCATS, and I know a lot of you probably are as well. So years ago, when NCATS first formed, almost 10 years ago, we developed a number of programs in repurposing. If you're interested in them, we have, of course, a website about early stage, late stage repurposing, technologies, collaborations to help with it, and go take a look if you're interested in that. One of the first things we did was to realize that at the time, back in 2008, there had never been a physical collection put together of every drug ever approved for human use worldwide. So we spent quite a while doing that, collected something originally called the NCGC, don't worry about that, what that name is, Pharmaceutical Collection. It's now called the NCATS Pharmaceutical Collection, but it just is that. So we needed a physical and electronic collection of every drug ever approved human use worldwide. This turned out to be a very complicated problem for reasons that you can read about in this paper. This paper reads a little bit like a murder mystery in some ways, because there's so many twists and turns at answering what seemingly, again, is a very simple question. But have a look, and it's in existence. It's been in existence now almost for 10 years. Actually started before NCATS was formed, its predecessor. And one of the things we found here, relevant to the open source mission, is that over 90% of the drugs in this collection are no longer under patent protection in one or more markets. This is a complicated thing to do too, because patents apply to individual compounds and formulations in various markets and for various indications. So one can't give a blanket answer to how many are off patent, but it's roughly 90%. And that is both an opportunity and a problem. So if you're interested in what we've done with this over the last 10 years, we just published a paper end of last year about a 10-year update on this program. This just shows you the countries and the states within the United States in which this collection has been used in collaboration with scientists all over the world, and the number of citations. And here's a table from the paper that talks about drug leads identified from NPC, that means NCATS Pharmaceutical Collection screens. So these are repurposing screens. And if you look down the drug list there, you'll see that a lot of these, most of them are off patent. They're generic drugs. And you can see the references there if you're interested. And we decided we needed to put this also in the typical kind of pharma Gantt chart phase of development sort of format. So this gives you the drug, the indication, and what stage it's in. And you'll notice that most of these, even ones that were discovered a number of years ago, have not made it to even clinical trials, much less approval. And that brings us back to the original question, well, if this is so much more efficient and effective and can treat currently poorly treated or untreatable diseases, and you look there on that list, you'll see them, what's the problem? Why haven't they advanced beyond the phase where we work on them? Because, of course, we make the information available to everybody, or we make all our data freely available to everyone. Before I come to the answer to that question, because I think it's what partially the Open Source Pharma Foundation is trying to address, but also what NCATS is trying to address. I want to give you a couple of other examples of things that we're doing to attack this problem in somewhat different ways. So also about 10 years ago, we started another program called the New Therapeutic Uses Program. This was a completely different approach. The approach here was this is a collaboration among NCATS, pharmaceutical companies which have these drugs, and researchers. And the idea was that the pharmas had drugs which had not been approved for the most part for any use. They had gotten into people, had been safe for their original indication, but had either failed for their original indication, no efficacy, or had been deprioritized for business reasons. So here are potentially, phase 1, phase 2 ready drugs, which a lot of work has gone into, and from a pharma perspective, they're non-performing assets. They're not producing any revenue. From a researcher perspective or from a medical perspective, these represent a golden opportunity to find a new use for these largely developed drugs. And what NCATS did was provide the asset information. We had template agreements that we developed that greatly sped the negotiation time, which can torpedo these kinds of projects very effectively. And we funded the clinical trials. And here's just an example. Again, this is all on our website, but we listed previously confidential information about these experimental assets. We call them assets because they're, in some cases, they weren't just small molecules. They in some cases were other kinds of biologics. On our website, And that was a real breakthrough for us and for the pharmas. And then solicited new ideas for them from scientists all over the world. And here's just an example. This is a Src kinase inhibitor developed by AstraZeneca, and it actually was repurposed there multiple times through this program for all kinds of other indications, non-cancer indications. And so this is not a screening effort. It's so-called rational repurposing. That is, the investigators had an idea of a disease, a new disease in which a member of the Src tyrosine kinase family was implicated. So this Src inhibitor, multi-Src family inhibitor, might be useful in treating one or more of those diseases. So it wasn't a screening approach, and most of them weren't off patent. They were on patent. They were collaborations with pharma companies who would then take these over if successful and complete the clinical development and marketing. One of the big things we did, which the open source pharma could certainly think about, because it's another one of these non-scientific or social scientific things that can torpedo projects. I think you're all familiar with the fact that if you put a bunch of lawyers in a room together to hammer out an agreement like this, it could take them 2 years, literally, to come up with a collaborative agreement. And people, I don't want to be too dramatic here, but it's true. People with diseases die during the time that we are negotiating agreements to do an experiment. And that, in our mind, is just unconscionable. And so, but nobody was working on this problem because it's not viewed as a scientific problem. Well, it is a social science problem. So we spent quite a while working with our private sector, our pharma colleagues, who were wonderful to work with, I must tell you, throughout all of this process, to develop these temple agreements that shortened the negotiation time by about tenfold, from 2 years to about 2 months. The next thing I just want to tell you about briefly is something that you might find helpful. It's not directly relevant to repurposing, but you may find it helpful in your own thinking, your own work, as well as in education. This is something called the 4DM map, the drug discovery, development, deployment map. This was something created by a group of us at the National Academy of Medicine. It's part of the National Academy of Sciences, Engineering, and Medicine here in the U.S. About half these folks are in pharma and half are in nonprofits and academia, if you look at the author list there. And the problem here was that most stakeholders, we say many, but it's truly most stakeholders, including policymakers, importantly, at least in this country, and I'm guessing in yours, are unaware of where drugs and devices come from. The complexity and difficulty of this process. There is a myth out there that this is really simple, and why do we have to have incentives? Why do we have to work on this process as a science? And of course, all of us know that it isn't. And we realize that one of the problems was that there is no accurate portrayal of the translational process that we could use to both re-engineer the process, right? So if you're going to re-engineer a complex process, you got to know where the rate-limiting steps are. Think about this as a biochemical pathway. For those of you who are biochemists, you want to know where the rate-limiting pathways are, because that's what you want to work on, because that's what's going to increase your efficiency and yield. And so there's a bunch of papers we published, and you can look at them, but I just want to show you what we did here. So you've all seen this chevron diagram. I showed it earlier. We tend to refer to this as a pipeline, but it's a pipeline that only allows 0.1% of what goes into the beginning to come out the end, and it takes 15 years to go from the beginning to the end of the pipeline. It's about the poorest pipeline you could possibly imagine. And we realized that using this in educational purposes with policymakers and scientists was really doing ourselves a disservice, and them, because they believed that it was this simple. And we said, okay, so we're going to make a map of really what it is. And this is actually what it looks like. And this is a simplified view. What we did here was to take each of the colors in each of those pieces of the chevron on the left and blew them up into multiple stages, and how they're connected, and the recursive nature of this. And then after that, we created a Google Map version. This is an interactive version, electronic version that is far more complicated. That's all on our website. So I use this both, we use this here, both to figure out where we are and where are the steps that we need to optimize the most. There's a traffic version of this, if you will, red, green, yellow, like a Google map on our website, on the papers as well. But also I find it very useful to use for policymakers, because if you show this to somebody and you say, well, you know, our basic science colleagues are in the upper left corner and they think they're done, and they've done really important work, but that's just the beginning, not the end, is extremely helpful. Okay, a couple of things about COVID before we finish the presentation part. We have viewed COVID in very much the way that Jay Kumar addressed at the beginning. That is, that it's not only a global pandemic, which to my way of thinking, requires those of us who are in this field to respond. I mean, we are not infectious disease people, we are translational people. And if we needed to respond to this, and I think we all felt that. And so we team up, as we do always, we teamed up with our infectious disease colleagues to do a number of things in the drug development, vaccine development area. And one of them was this open data portal. So what is this? It was a result of a realization that labs around the world all started testing drugs and doing repurposing as potential treatments for COVID-19, essentially simultaneously, last January, February, March. But per usual, results and methods weren't shared quickly. They were put in publications, et cetera. And even when they were, all the data that you really need to reproduce the data are typically not published, you know that whole story, and not easily available to the scientific community. So, of course, that limits their impact and delays how quickly we can bring treatments to patients. So we thought, well, gosh, why don't we use the urgency of COVID-19 to do what we always do, to run thousands of experiments, that is, testing all of these thousands of drugs, in this case against a suite of COVID-19 assays. And I must say, though it shows a multi-pipetter here, we actually don't use those most of the time. We use these things, which are big automated robotic systems. But run those across all approved drugs and make all the data immediately available to the world. So we established these open data principles where the new data is uploaded to our site as soon as it is quality controlled and validated. We do not wait for a publication. We don't wait for a bioarchive preprint. We put the data on there as soon as it is QC'd, and don't worry about the publications. They will come, but that we didn't wait. We wanted all of the data to be viewable, the positives and the negatives, and searchable in a way that if you're not a bioinformatics expert, you can still make sense of the data. It's very important. All the experimental protocols, and I mean protocols here. I don't mean general summary that we put in a methods section in a paper. We're talking about detailed protocols shared openly on the site so others can run them. And if they can't run them, it's our fault, not their fault, because we haven't provided enough data. And you can download all the screening results. This, in our mind, is how the system always ought to work. We have been unable to do this up until COVID because of the usual kinds of social science people limitations, but we've done it now with COVID. And perhaps one of the most exciting things is the communications that we've gotten from other researchers in the US and other countries who want to put data in here as well. A last example is something that you also might find useful. The problem we're addressing here is how do clinicians, particularly all over the world, who are not at a major medical center, for instance, share experience of treating diseases, in this case COVID-19, but really any disease, in real time. So we in the Food and Drug Administration here in the US, a bunch of scientists from FDA and from NCATS, created a mobile app that allows healthcare workers to share their clinical experience using approved drugs to treat diseases other than the ones for which they are regulatorily formally indicated. And so you can contribute data, you can see other people's experience, you can discuss on the app challenging clinical cases and treatment questions. It's available on the App Store and on Google Play. We've limited this to certified healthcare professionals because we really want them to have the science and data and evidence of sort of standards that all of us have. We didn't want this to turn into a version of Twitter. You know, those things already exist, and we know how unreliable they can be. And I just want to show you. This is the desktop version, but it looks there's a version of it on the mobile as well. We actually created this for all infectious diseases, particularly neglected diseases. And this went live in December, actually, right before COVID started. And so it's well timed. But you notice it's not just COVID, but things like adenovirus or trypanosomiasis or acanthamoeba. And I hope you'll go look at that as well. But if you look at COVID-19, you'll see here the number of case reports. And this is an old slide, so the numbers are larger here, but you'll recognize a number of compounds that you would have heard about, including hydroxychloroquine and azithromycin and interferon and methylprednisolone and lopinavir, ritonavir, etc. So we're hoping that these open efforts will serve as the nidus for behavior change in the community more broadly. I must tell you that I am cautiously optimistic that some of the changes that we've seen with COVID will persist, but I think we cannot rely on that happening without action. I mean, there's the biomedical ecosystem is remarkably homeostatic. And it will, I believe, go back to its previous way of doing things unless there continues to be a push to keep these really beneficial behaviors for patients, a push to make those continue. So it's something that I think the open source movement can play an important role in, and it's something I think that Jay and I might want to talk about now. So those are my comments. Jay, I hope you're back on the line. I am going to unshare. Have I unshared?
I think you've unshared already. Thank you so much, Dr. Austin, for your presentation. It was incredibly enlightening, and I think we all have so many questions now, not only about the work, but how to go forward and how best to use this open drug portal that you're speaking of. I think we're gonna start the Q&A, so for everyone to please place your questions in the Q&A box at the bottom or in the chat box, and I'll be reading them out for Dr. Austin to share his insights. So yes, again, thank you so much. That was really stellar and so interesting to see how this disconnect can really be addressed with open source methods. One of the questions is how can this open source portal and method also address the social science or the social behaviors that torpedo a drug repurposing project?
Mm-hmm. Well, Jay knows this because he was at a meeting that we had back in December where we spent 2 days exploring this. And like so many things, one has to look at the whole process to be able to answer that question. And it will not surprise any of us to learn that many of the limitations come down to money. So if there isn't a business case to be made, it's very hard for these things to get developed. And and so they died not because the projects died, not because they don't have promising science behind them, but because there isn't a company who can meet their fiduciary responsibility to their shareholders by developing this, because they can't charge enough money to recoup their investment. And so,
Okay.
So who drives this? Well, who pays for drugs? So you can't blame it on the pharma companies, really, because they're doing what they're supposed to do. It's the insurance organizations, it's the reimbursers, it's the healthcare organizations that pay for healthcare in the various countries. And oddly, they often they are not willing to pay for development of some of these treatments, even if it would save them down the road a very large amount of money, because they don't see that as their job. And in some ways, it hasn't been their job. And so that's what we're trying to figure out. We're starting with the people who pay, who are the in this country, Medicare, Medicaid, insurance companies, and working backwards. We had them at the meeting, and they didn't even know this problem existed. Interesting. So some of it's just bringing together the community, and people go, "Oi, oh, that's the problem." Okay, well, why don't we think of a new way?
Thank you. I'm gonna give the mic to Jay to ask the next question.
Well, Chris, thank you for a stellar talk, and I think I get the prize for the least timely system failure. But we had foresight, and we had Alina there as the backup to do the introduction. So thank you, Alina, and thank you, Chris, for bearing with us. Again, a real treat to have you here, Chris. And I wanted to ask about, one of the reasons why a lot of people on this call and around the world are interested in access and affordability.
Yeah.
So the advantages of drug repurposing are twofold, really. One is speed, but the other potential advantages is price.
Sure.
And is there something that people who are uninsured in the United States or who are in Kenya or in India can afford? Sure. And it's a very basic question, but I thought it might be worth touching on. Your outfit very presciently did a whole workshop on off-patent drug repurposing.
Yeah.
And it's a basic question, but maybe you could explain to the folks on the call here, why off-patent, and what is the potential economic advantage there to the health system and to the patient?
Yeah. Well, first of all, what do we mean by off-patent? When a drug reaches the end of its patent life, then it's open to generic competition. So in general, especially with small molecule compounds, but not so much with biologics, the price comes down dramatically. So the cost could come down 80, 90, 95%. And interestingly, in this country, about 90% of prescriptions are written for generic drugs already. The cost is still, total cost is still accounted for mainly by the branded ones, because they're 100 to 1,000 times as expensive. But there is a lot of interest in those off-patent drugs. The problems range from who funds the clinical trial to show that this off-patent drug is useful in this new indication, because the companies won't do it, because they can't recoup their investment. The NIH, at least, for the most part, does not see this as their job. They say, well, the companies should do it, and the companies say, well, NIH should do it, so nobody does it. For foundations, this is really hard. These can cost 10, 20, 30, $50 million, one trial, which is just out of reach for almost all nonprofits. So just generating the data that FDA or another regulatory agency would need is exceedingly difficult to do. And though we're a government agency with a quite generous budget, we don't have the money to do this either. In a general way, we do it in the ways that I described, but not for the whole problem. The other problem, it sounds really boring, but it's another one of those things that kills projects, which is labeling. So what the heck am I talking about? Labeling. Well, at least in this country, physicians pay great attention to what the drug label says. That's the big long piece of paper, usually about 4 inches wide, that you get when you go to the pharmacy and you never read, that has all the side effects and all that kind of stuff, pharmacokinetics. And on that are the indications, the diseases, the conditions that FDA has said you ought to treat this, that this drug treats. The only people who can change that label is the company that created the drug in the first place. And if the company that created the drug in the first place no longer makes it, because it's an off-patent drug now, then there is no company to change the label. So the FDA says it's got to be on the label. The company says we don't do this drug anymore, so we're not going to change the label. It would cost us a lot of money, and we're not going to do the trials. So the physicians say, look, I need some coverage from FDA or somebody else to say that this drug is actually useful in this disease. And so you got to have the FDA change the label. And again, it's one of these things where everybody's pointing at somebody else. And then there are reimbursement problems. It's astounding in this country. And I'll give you an example from another public health crisis. There are generic drugs available for opioids, which are not being used, and for alcohol disorder, that are not being used because they are produced on such a narrow margin that the manufacturers have gone out of business. And so the drugs are frequently not even reliably made because the profit margin is so narrow that the manufacturers don't exist. So it's a multi-headed problem. It's a regulatory problem. It's a reimbursement problem. It's a manufacturing problem. It's an incentive problem. I think everybody wants the same thing, but it's one of those problems that fits. And Jay, you have a background in economics, I know, so you'd be familiar with the tragedy of the commons problem. That's really what this is. The tragedy of the commons is something that's everybody's problem in general, but it's nobody's problem in particular, so nobody solves it. But that's the problem, and it's the open source problem writ large. Outside of repurposing, has a lot of those issues as well. Now, this is solvable, right? Because if you look at an organization like DNDi, Drugs for Neglected Diseases Initiative, that's funded by the Gates Foundation at the level of hundreds of millions of dollars a year, has done remarkable things at repurposing off-patent drugs for neglected tropical diseases. And so it can be done. But again, they're getting large amounts of money from the Gates Foundation, who are focused on neglected diseases, which is great, but it's a narrow window.
Thank you, Chris. We have several questions, and we have sort of 7-8 minutes left in our window. So I would propose I might ask a combination version of the questions, and then to the extent that's possible, Chris, you can address them. And if people, I think we'll let you go right at the hour. So because we appreciate your time. So the 4 questions are, one is, in India, there's a lot of herbal medicine, and what can be done with that? The second is a detailed question about your Cure ID platform, which is wonderful. It's crowdsourcing, clinical ideas. And is the data from that, and this is a little bit of a tough question, is the data from that, are there plans to release that to the public? Mm-hmm. And the third question is, many of the existing drugs are optimized for specific targets. So what does that optimization do in terms of off-target? I mean, is it still possible to get off-target effects? Mm-hmm. I think we know the answer to that. We for example, OSPF is in phase 2 clinical trials for metformin, which is optimized for diabetes, but we're trying it against tuberculosis with the government of India.
Yeah.
And then the last question is about drug combinations and whether NCATS works in that area. So we've
Yeah.
got herbal, cure ID, optimization, and combinations.
4 very simple questions. Okay, herbal. We've been working on natural products, another word for herbal therapies, for as long as I've been here, and actually back at Merck too. That area is limited by technical issues as much as anything else. These plants and animal product, corals, marine natural products are mixtures. And so figuring out what the active agent is in the mixture turns out to be an exceedingly hard technical problem. It's a whole nother seminar. It's actually quite interesting, but it's a technical problem. These things also tend to be, because they're made by Mother Nature, not by human medicinal chemists, they tend to be very, very complicated. If you've ever seen the structure of erythromycin, you know what I'm talking about. Or cyclosporin. They're very, very complicated molecules that are very difficult to make, and so end up being extremely expensive. And so they end up often the drugs that are identified, if there is an active drug identified in an herbal therapy, then it is patented, and the way to make it, even more importantly, is patented, and then it's very expensive. It's not generic. Now the herbal preparation is, of course, is available all over in India and this country. The problem is that and you probably all know this, that what's in those changes over the course of the year, depending on the life cycle of the plant, and it changes in the ground that the plant is growing in. So you could take the same plant different times of the year in different counties or different parts of the same country, and one will have a good effect and the other one won't, and we have no idea why. And you just can't have a drug that works that way. So that's why people have said, well, we got to isolate the active ingredient and then make it. So it's very tantalizing. I often refer to natural products as made by or herbs as the great medicinal chemist in the sky. That's, Mother Nature has made these things. But it's a technical problem as well as it's a technical problem as well as an IP problem. The Cure ID issue, we're working on that with the FDA. They are, you can understand, they do not want to, they don't want to incentivize the use of approved medicines by folks who wouldn't be able to interpret the data. So that's why they want to keep it limited to healthcare providers. Now, frankly, all you got to do is just check a box that says you're a healthcare provider and get access to the data. But it certainly is something we would like to do. We'd have to put caveats all over it to say, these are anecdotal observations, these are not clinical trials, et cetera, et cetera, et cetera. But it is very useful. The off-target issue is a really interesting one. So 2 ways to answer it. First, you did already, and I mentioned it in the Sark kinase. Because Mother Nature is the original repurposer, and she's a repurposer in targets as well as in natural products or the small molecules that govern our living systems. The same target, the same gene product is used over and over and over again in multiple different settings, in multiple different organs and different cell types and different stages of life and disease. So even without optimizing, the same target could actually be a good and the same drug on that target could be useful for multiple different diseases, and we've seen this before. The other way to think about it is something that I was told is a very important adage that was said to me by a wise medicinal chemist at Merck when I was there. And you may know that Merck is a very big chemistry company. And when I first got there, he said to me, Lawson, you got to know that every drug has 2 activities. I was there with my pencil, right, the great sage is going to give me this this pearl of wisdom, and he said, the two are the target you know about and the target you don't. And and it sounds silly, but it's really true, right? So these compounds are never monospecific. You can try to optimize them, but Mother Nature's got 500,000 targets in different, in the human body alone. So they're all going to have off-target effects because of the non-specificity and dirty pharmacology, if you want to talk about that, of even the most modern drugs. The older a drug is, the dirtier it is, the more off-target effects it has. But the other implication to your question is, another adage that I learned at Merck, which is true, is the best way to come up with a new drug is to start with an old drug and optimize it. And we see this a lot too. So repurposing, even when you can't go directly into a human with that compound, it's a great place to start for med chem optimization. Combinations, what a great question. Yes, we have been very interested in combinations for years, 15 years, and actually have developed a very, very cool technology to do many-to-many combinations. The big question is 3,300 compounds times 3,300 compounds in 10 different concentrations. That's what you'd want to know. The problem is that number gets into the trillions very quickly. But we have developed a way to do subsets of those and winnow down combinations looking for both synergy and antagonism. We're doing it right now in COVID, as a matter of fact. And a lot of people say, well, FDA doesn't like combinations. It's not true. If you go to FDA, and they're very close colleagues of ours, they put out guidance about combinations. I mean, there are some special considerations, but it's not as bad as people would think, and so we also have information on all that on our website too, if you're interested. And something I should say for everybody that's on the call, in case I forget to say it, is everything I described and everything at NCATS is a collaboration. Very, very little of what we do at our place we do alone. These are almost always projects that people somewhere in the world come to us with and say, I have this new target, I have this disease, I have this something, and I don't know how to advance it. I know a lot about the disease or the target, but I don't know anything about drug development and all that stuff. Can you work with me? And that's a wonderful thing for us, because we don't know anything about the disease. We can't possibly know everything about 7,000 diseases, right? So we rely on our colleagues to bring us that. We also rely on our colleagues, like you, Jay, to bring us knowledge of how other systems work and what the needs are in other countries, because we don't know. So so people on the phone or on the webinar, if you have these ideas, we want to hear from you, right? Because everything we do is a collaboration. We don't start unless we have somebody bring us a problem, because then we know it'll be useful. And for some of you are saying, well, how do they do that? If they have tenure and all that. We don't have tenure here. We don't believe in tenure. We don't have principal investigators. We don't work the way the rest of academia works, because we think it is antithetical to the behaviors which are needed to change this field.
Chris, that was wonderful and rousing. We have a few more questions, but I think we will let you go fight COVID and the other 7,000 diseases. Yeah.
What we might do, and I've done this before, is if you or Lena want to write up these questions and send them to me, then we can just post the answers on your website or something, whatever you want to do. I'm happy to do that.
Oh, that's exceedingly generous of you, Chris. I mean, that's to use the old Yiddish word, you're a real mensch. And so thank you for that. And it's really an honor to be able to spend a little bit of time with you, to hear from your experiences and your scintillating intellect and your big heart. So thank you for doing what you do. I think the world needs you. The world needs all of us to collectively solve this problem. And we express our gratitude. Thank you.
Well, back at you, Jay Kumar.
Thank you so much, Dr. Austin. It was a total honor, and I want to tell everybody that's still on the call, thank you so much. We will be sending your questions to Dr. Austin now that he's so graciously offered, and we encourage you all to go on to osbfound.org and the NCATS NIH website, which is www.ncats.nih.gov for more information. And reach out to us on Twitter or Facebook, or, you know, there's a million different ways. So keep in touch, and thank you to everyone. We had almost 150 participants today and more than 30 questions. So thank you again. We are incredibly grateful and looking forward to working further.
Wonderful.
Thank you. Thank you.
All right. Thanks for having me. Take care, everybody.
Bye, everyone.
Bye-bye.